is shortly past 3 o'clock, so I think we should get started. Welcome everyone, and thanks for tuning into today's Distinguished Lecture Series seminar of the Max Planck Institute for the Science of Light. This is the last seminar in this present series of DLS seminars. I want to extend a particular welcome to our guests who have registered for the DLS and are joining from outside the Max Planck Institute. The DLS seminars are the main high-profile scientific event of our institute, and it is a means to engage the most important and influential colleagues in any area of the science of light. So today it is my pleasure and honor to introduce Jürgen Popp, Professor of Physical Chemistry at the Friedrich Schiller University in Jena, and is also the Scientific Director of the Leibniz Institute of Photonic Technology in Jena. Jürgen is best known for his great work in biophotonics, where he has published more than 800 papers, and in particular on Raman spectroscopy in its many different guises and uses. Jürgen has received many awards and distinctions, including the Pittsburgh Spectroscopy Award, the Kaiser Friedrich Forschungspreis, the Ralph Dahrendorf Award for the European Research Area. And what I found particularly interesting and telling is that he's an elected member of both the Optical Society of America, as well as the American Institute for Medical and Biological Engineering College of Fellows. So this highlights that he does not just develop optical technologies, but he has the drive and desire to translate that into medical research and clinical application. So this is also very fitting for, in particular, the area of research of our institute that's geared towards maybe a physics of medicine or physics and medicine area. So very much looking forward to your talk. Before we get started, two technical announcements. The seminar will be recorded, so please be aware that by participating, you implicitly agree to this. And it would be great if you could keep your mic turned off during the presentation so that we're not interrupted. If you have a question, please either type the letter Q into the chat space or the question in full if you want to. And then I can ask you to unmute and then you can ask your questions. We can do that for very urgent questions throughout the presentation, but most of those questions will then be asked at the end. So enough from my side. I want to hand over to Jürgen. The stage is yours.
Yeah, many thanks for the invitation, also for the nice introduction. And it's a pleasure to give the presentation here. Even though I would rather prefer to be in Erlangen, not just giving this presentation, but also seeing the institute, talking to the people and having a real audience in front of me. But nevertheless, these are the circumstances at the moment, and we all hope that this will change quite soon. And you may see from this presentation that we are also working in this field of infection diseases. And what is driving us is, yes, we like to do fundamental research. We like to start from ideas, going to the application, but our interest is also to go to the instrumentation. Very often other groups or for other research institutions, this is boring, but I think this is also something which is quite important when you think about new technology, that you also think about how can I implement this in a clinical setting, how can I implement this really where this technology is needed. And therefore the title is quite simple. It's Translational Biophotonics from Ideas to Instruments, and I hope I can give you some insights about our philosophy. Nevertheless, before I start, I'd like to give you a little bit of a brief introduction where we are from. Certainly you should know Jena and Erlangen, they have a close collaboration. The universities have a close collaboration, but also various institutes collaborate quite nicely. And here you can see Jena, it's a quite long elongated valley, and I think it's quite well known because of these people. On the left side, you can see Carl Zeiss, Ernst Abbe, and Otto Schott. And why I'm showing this, not just to mention that we are talking about the optics valley in Jena. No, this is somehow also what is needed in order to think about translation. First of all, Carl Zeiss started his company, but at that time, more or less making microscope was by trial and error. There's even a word for this, it's Brübeln. You take lenses, you put them together, and you are trying to make something like a microscope. Then Ernst Abbe, the mathematician, the physicist, came into the field, and with him, more or less, it started that you can do things by design. You look for the resolution, you look for the optical design. Nevertheless, even Carl Zeiss and Ernst Abbe could do a lot of things, but something was really missing. And this is then the appropriate material, glass, and therefore they were quite lucky and happy that they could convince Otto Schott to build the glass manufacturing not somewhere else in Weimar, but in Jena. Nevertheless, this is the technical part. What is also driving, the development of a new microscope is the request, the unmet need. And this unmet need is defined, for example, here by biologists like Schleiten, like Haeckel, and they want to have specific performance of those optical devices. And I think this is why or what is needed at the very end, if you think about fundamental research, and then this is going to the translation. You need more or less both sides, the technical part, but also the application part always in mind. Well, Jena is more than this now. We have the university, we have the university hospital, we have the University of Applied Science. And here you can see the Beutenberg campus. This is something which has been built after reunification. But also we are quite strong in industry, optical industry, but also biotech industry, which is more or less really pushing towards new ideas, new instrumentation. And just give you a little bit of a view on the Beutenberg campus. We are now zooming in, and this has been started after reunification. In the meanwhile, we have more than 10 research institutes, two buildings for new companies, spin-off companies, and then you can see, or not you can't see, but we have something like 3,000 people, scientists and technicians working here just in this research campus. And what you can further see is if you go a little bit closer to our institute. So this is the Leibniz Institute of Photonic Technology. So you can see this is our main building. We have a clean room facility. I will show you later on what we can do there. We have a laser application facility, and back here we have our fiber drawing tower. And this allows us to build our own custom-made fibers. So just to give you some figures of the institute, we have something like 370 up to 400 employees, 100 PhD students, something like 220 peer-reviewed publications every year, and the total budget is around nowadays 26 million euros. And half of the funding is coming by third-party funding. Well, this was just a little bit an introduction into the institute where I'm from. Now I'd like to come to the topic. So I'd like to start with this picture. You have this situation. For example, you are sitting in front of a physician. You are talking about that you are not feeling well. You have somehow fever. You have a cough, and after something like 20 minutes, a half hour, then you are leaving with a certain prescription, and this prescription is saying you get an antibiotic treatment against, let's say, Escherichia coli. Now the question may arise, how does the physician know that I have this infection? How does the physician know, is this really a viral, is this really a bacterial infection? Is this not a viral infection? Why does she know that the infection or this antibiotic treatment will be helpful? May I have a resistance, or my bacteria will be resistant against the antibiotic? Well, and these are open questions, and this is what we are trying to answer. Is it possible really to get in a very short period of time by taking, for example, a urine sample, a blood sample, all this information about what is causing these symptoms. Is this really an infection or is something else? And this by using some biomarkers, cells from the patient, and then using optical technology. So this was the idea behind this. So what we'd like to provide is a precise diagnosis before a target therapy will start. And as I started with the first example, I will go on with the topic infection and also talk a little bit about sepsis. And later on, I'd like to come to another topic where we talk about cancer, IBD, and maybe also if time is, if there is enough time, then also to talk about how we can use the eye as a window to the body. Well, what is needed here in the case of infection for an optimal and personalized treatment? Well, what you need to know is, in this case, you need to learn something about the host response. What does host response mean? It's quite simple. The symptoms like fever, which may correspond to an infection, can also correspond to something else. So we need to learn something about, are the symptoms really coming from an infection? So the host response is something which is quite important. Then if it's really coming from a pathogen, then we should identify the pathogen. And if it's a bacterial infection, then we should know about the infection, about the antimicrobial resistance, if possible. And then again, as I said, the antimicrobial resistance is then something which is quite urgently needed to be identified. Well, what is the host response? And then we have to look at the role of the immune cells during an infection. And we have here different immune cells like the basophil, the B cells, the T cells, the neutrophils, monocytes. And those cells, they interact, for example, very nicely with the infection. The microbes, like it could be a fungi, here like Candida albicans, it could be a virus, it could be a gram-positive or gram-negative bacteria. And what happens is that those cells give you a certainly cellular response. Yeah, there will be some changes in this response, not going into the details, but what you can somehow imagine is that if there are some molecular changes occurring, then those molecular changes we should be able to target, or at least to monitor these responses, to change, to see what is going on by those interactions. And what we try, or what we are using in order to learn something about this interaction of this, let's say these white blood cells, these leukocytes with the pathogens, is using Raman spectroscopy. Why? Because Raman, I would say, is one of the most simple techniques. You don't need too much. You just need your target. You need your light source, like a laser. And here you are quite flexible. You can use any type of laser from the UV up to the NIR. So you will get certainly different responses from the molecules. You can hit electronic resonances. You can go for resonance Raman. You can go to deep UV resonance Raman. Nevertheless, what you get at the very end is something what we call a molecular fingerprint information. Well, when we started this quite naive, because not really knowing about microbiology. And what we realized is at the very end that just looking at the Raman data is not really sufficient. What you need to do is you need to work really on the data analysis. And then we are opening the field of machine learning, deep learning, artificial intelligence in order to really interpret this complex Raman data. Because what you are doing essentially is you are probing the molecular composition of a biological cell. And if we talk about, for example, we want to identify different bacteria, or we want to identify the host response of neutrophils interacting with some pathogens, then you can imagine that the underlying chemical situation of the various cells will be quite similar. And in order to retrieve these small changes, you need this machine learning, this mathematical approaches in order to get this information out of the Raman data. Well, this is when we started this journey in 2003. This was when I was appointed in Jena as a professor at physical chemistry. Then we were using this argon ion laser or krypton ion laser and having a large microscope back here. Then we had our first portable or miniaturized laboratory set up. This is a German project funded by the research ministry. It was called OMEP. And then in 2010, for example, we were moving then already to the clinic, to the hospital. We introduced here the Bioparticle Explorer. This was a joint venture development with a company in Berlin, the RepID, which is now the company Mibek. Well, when we talk about host response, then what we are doing more or less is we get from the patient something like 500 microliter blood. As you can see, this is not really a lot of sample. Then we have our quick sample preparation. So we isolate the white blood cells, and this we can do by lysing, destroying the red blood cells. Then we put them on a calcium fluoride slide, and then we could do our Raman measurement. So this is a quite simple approach. And then there are various ways you can do. When we started this, we were measuring point by point over the cell. And what you get is, for example, if you look at a neutrophil or a T lymphocyte, you get your molecular fingerprint information. This is a typical Raman spectrum. And you see some distinct features, but it will be very difficult just by judging by eye in order to say, oh, this is a neutrophil and this is a T lymphocyte. Yeah. So, but what you can do is you can use, for example, a very simple approach. You are looking, for example, for the DNA band of your cells, and you compare this with the cytoplasm, and this allows you, for example, here you take the band of 785 inverse centimeters, and you plot this against the 2030 inverse centimeters, and then you can see where the nuclei is. And just by this simple approach, you can easily see, oh, this cell is, for example, a lymphocyte, this is a monocyte. You take into account the size, and here you see what where the nuclei is, and here you can see the spreaded nuclei. So these are the neutrophils, and this way, for example, you can easily use Raman information in order to identify the different cell types, and this in a complete label-free way. Well, what is quite important when you look in publications working with cells, that there is very often just a few cells being investigated. The reason for this is that Raman is not very fast, and if you want to measure a single cell and you really scan over this with something like 1,000 points, and each point takes something like 1 to 5 seconds, and then at the very end you end up with let's say a half hour for a single cell. But what you can see, if you measure just, for example, 100 cells, then you can see the distribution, and you can easily realize that with this approach, if you come up with this point and this point and this point, at the very end, you will judge that the distribution of your different white blood cells is completely messed up. So what you need in order to get something which is really reliable, which is significant for what you are finding, you need to measure something like between 1,000 to 1,500, 2,000 cells. Here you can see now the cells are converging, the signal is converging, and then you have a nice prediction of the various fractions. So this means if you just measure a couple hundred cells, then this result is not really significant. For what is important, you want to measure something on the patient. You want to see what the health status of the patient is, yeah, and there you will have changes in the distribution of the neutrophils to the leukocytes, to the lymphocytes, and so on. So therefore, you have to be very careful. And here you can see now, for example, what we found with Raman, and we compare this with the classical clinical readout. Here we look at the neutrophils, the monocytes, and the lymphocytes, and you can nicely see that the Raman readout is nicely fitting to the clinical readout. Nevertheless, here we measured a large set of samples, something like 100,000 cells have been measured for this type of sample. How do we do this? Normally, we cover a slide with cells, and here you can see this cover slide. It's 5 millimeter versus 3 millimeters, and this is completely covered with cells. In this case, we measured something like 24,000 cells. This was one of our first approaches. Here we need something like 6 hours for this entire slide. Then you can see if you don't need to measure something like 24,000 cells, but if you need to measure something like 1,200, 1,500 cells, then you can really shrink down the time and you can go from patient to patient. You can measure many, many patients. And here we have developed the Raman approach, which is doing this completely in an autonomous way. And what is quite important, we rethought Raman. Raman, normally, if you're talking to a physicist, like my mentor, Professor Kiefer in Würzburg, he was always aiming for highest spectral resolution, because then you can take a lot of information out of your Raman data. But if you look at the Raman data from a biological cells, the features are quite broad. There is no reason in order to go really for high spectral resolution. And this also means you can save a lot of time, because you can open the slit, you can go to a less good grating, and this means you can put a lot of light, Raman light, into your spectrometer, and this way you can decrease the integration time on your detector, and therefore you can measure much faster. And this was one of our approaches and successes in order to go really for large number of samples, and then getting really reliable data from one single patient, for example. So let's give you some results. And what you see here is one of our studies, which we have done by spiking experiments. So what we did is we isolated from patients the white blood cells. Then we spiked them, these cells, with bacteria, and this bacteria with different gram-positive, gram-negative bacteria, with virus particles as well as fungi. And then we looked at the host response. The approach was quite simple. We isolated the cells when they were spiked. Then we washed them so that we removed all this bacteria again. So we were just looking at the cells itself. And then we were interested at the activation state. And what you can see here is one similar or simple approach. What we did is, after our preparation of the Raman data, background correction and all these things, then we did, for example, here an investigation with a principal component analysis in combination with a linear discriminant analysis. And here you can see that we can nicely separate the infected neutrophils from the control. Control means these were the cells without any infection. And you can also see the loadings over there. You get some spectral information, what is going on the molecular level. So this is not really completely black box. And then if you go for the fungal infection again, you can also compare this. You see here different features compared to the bacterial infection. And what we at the very end succeeded is, when we take the neutrophils, infected versus non-infected, then we can easily, with an accuracy about 90%, we can differentiate between those neutrophils which has seen a pathogen from those which have not seen a pathogen. Then if we go for the infected, then it was easy to differentiate between fungal infection or a bacterial infection. And if we went then further with the bacterial infection, then we even were able, with not that high accuracy, but around 80%, which is already quite nice. We can even distinguish between gram-positive and gram-negative infection. And this you should keep in mind, because just by looking at the white blood cells, if you can really say the person has an infection by a gram-positive or a gram-negative, then this allows you at the very end also to look for the appropriate antimicrobial treatment. Yeah? So this combination of looking at the white blood cells would give you at the very end also a technical approach, and this I will show you later on, to the antimicrobial resistance. And then this is much easier by combining two different techniques by Raman spectroscopy. Well, now you may say, nice. But what happens if you have real patients? And here we had a clinical trial in Jena running, and a similar trial we had in Athens with our device. And in essence, this device has been just controlled by a physician. So there was no technical people more around. So we have used our Raman device or developed the Raman device in such a way that it is working quite autonomously, and we can remotely control all the function also from Jena, while it was in Athens. But now I'm showing you some results from the data we achieved in Jena. So we had here 24 patients included which have a styral inflammation. So this is not related to an infection. Then they had an inflammation which was caused by an infection, but this was an infection without any organ dysfunction. And then we had included 18 patients which have a sepsis according to the sepsis 3 definition. So the question was now, what can we learn by using Raman spectroscopy? And here you can see again the experimental flow. Quite simple. Lysis of the white blood cells, washing, putting them on a cover slide, calcium fluoride, then doing the Raman measurement. Afterwards, we do the Kimuna staining in order to see if our finding is correctly, if we identified the cells correctly. And what you can see here is now the receiving operating curve, the ROC curve, where we looked for the infection detection. And here you can see, for example, plotted 1 minus the specificity against the sensitivity. And if we would be completely correct, then you can see, then we would have a curve like this. So what do we see here? If we look at Raman, then you can see that we have an area under the curve, which is around 80%. The specificity is around 60%. And if you go for the biomarkers, biomarkers will be the standard biomarkers like PCT or SUPA. And there you can see that the Raman and the biomarker has more or less quite similar values. And what is quite nice, if we now combine Raman with the biomarkers, then you can see that we have a very nice values, the sensitivity as well as sorry, it's not the sensitivity, the balance accuracy, and the area on the curve is around 92 or 90%, which already means that by these two techniques within a hospital, we can be much more accurate. But the Raman data itself already shows you if you are having no chemical, no clinical chemistry, and normally a hospital does not have this clinical chemistry, then even if you would have the Raman, you would be able to get with a very simple approach the same values like with the biomarkers. Then the question was about the sepsis detection. Can we use Raman for the sepsis detection? And what you see here, if we look at Raman by himself, then we have here this area and the balance accuracy and the area under the curve. We have here around 71% and 82%. And if you combine Raman with the biomarkers, then we are at 92% and 99%. And if you go for the QSOFA, which is now essentially these values from the patient, for example, high blood pressure, fast heart beating, and so on, these biomarkers, the physical biomarkers, then they are less good or almost comparable with the Raman. But if you combine everything, then we are quite nice at 92% and 99% area under the curve. And this shows you how powerful the combination of Raman with biomarkers is, but even Raman by itself is already quite successful and quite nice.
Can I ask a question?
Sure.
Okay, just so that I can continue understanding. The Raman data that you have, or the judgment that you make based on the Raman spectra, was that just comparing infectious versus healthy in some kind of a general way, or are you actually after a certain type of lipid or a certain bond, or do you have a clear kind of line that you look at, or do you look at the whole landscape and establish a certain catalog like you showed that something is different?
So first of all, these are our cohort. These are the patients which have been included. Yeah. So we have people with a sterile inflammation, with infection, without organ dysfunction. So this is where the sample came from. And from the Raman data, we took the entire Raman spectrum as, so to say, the biomarker.
So again, you don't really necessarily know why a certain part of the spectrum is different, exactly the biochemical kind of origin of that part of the spectrum, but you establish in an empirical way that this is how it's different when it's, for example.
More or less, this is the way it is. What we can certainly do, as I have shown here, we can get somehow a molecular understanding what are somehow the features, yeah? But it's not possible to really pinpoint this down to a specific protein or to a specific lipid or whatever, because this is not doable with Raman spectroscopy. What you have here is always the overlay of many information, and with the mathematical approaches, we retrieve then the underlying information, but more or less as a phenotypic information. Yeah? It's not like that we can go for a specific molecule. This would be great, because then with mass spectrometry, you can go for this simple molecule or this complex molecule, but this is not what we can do by Raman spectroscopy. So the Raman spectra, if I go back to a specific Raman spectrum I have shown to you, just a second. Where is it? Here. If you look here, you can see they are quite similar, and the underlying features is always a combination out of various molecules. And to get through to a specific type of molecule is not possible with Raman, at least by these biosamples, what we are looking at the moment. Yeah.
Thank you.
Is this answering your question?
Yes. Thank you.
Okay. Good. Then the next stage was, can we really go for this question? We get a sample and we can tell the patient, we can tell the physician, this is a patient having an infection by a virus, or this is an infection caused by a bacteria. And there we get some samples from the emergency department. Again, something like 500 microliter blood has been used. The procedure was the same. We were using our high-throughput Raman spectroscopic analysis. So this means measuring something like 1,500 cells in a very short period of time. And what you see here are the individual patients which we have measured. And this where we identified this is a viral infection, and this was a viral infection. This means there needs to be an antiviral treatment. Here you can see the patient having a bacterial infection, so they need an antibiotic treatment. Unfortunately, there was no patient present with some fungal infection. Again, here you need another treatment, an antimycotic treatment. Nevertheless, what we can show is that with this Raman spectroscopic information, we can go for a diagnosis-driven therapy, at the very beginning already saying what type of treatment needs to be anticipated for this patient. Well, now, since we know that we can use this spectroscopic approach for the identification of the host response, now the question is, if we know this is a bacterial infection, can we also learn something about what is causing the infection, what type of bacteria? And can we also learn something about the antimicrobial resistance? And let me first introduce to you this dilemma of antibiotic resistance. Well, this is here a map of Europe, where you can see the antimicrobial resistance of the bacterium Escherichia coli against the third generation of a specific type of antibiotics. This is the third generation of cephalosporins. And what you can see in 2002, there was almost no resistance. What does it mean? In the case you would have an infection by E. coli, then the physician would easily say, okay, then we take this type of drug, and this would be correct. So the physician, when I started my presentation, is more or less right, but not anymore. But in 2002, it was the correct way in order to come to a treatment. And this is just because of knowledge. Yeah? If there's a urinary tract infection, this can be caused by E. coli, or mainly it's caused by E. coli, and then I would take this drug. But time changed, and you can see that we get more and more resistance. And this means in the case you will have a resistant bacterial infect, or your bacterium will be resistant against this type of drug. Like, for example, in Italy, you would have already a probability of 50% or up to 50% this drug will not work anymore. And if you have a severe nosocomial urinary tract infection, which you can get in the hospital, for example, because of the catheter being introduced into your urinary tract, then this is causing something severe, a severe infection. And if they treat without knowledge, then it could be that the treatment is not really being successful. And this map is from 2016. I will show you, I could show you many other maps. If you are interested, then just go to the European Center of Disease Prevention and Control. Then you can see the various maps, and you will figure out there are many maps which are already quite red. And this means we are close to the border where we have no functioning antibiotic anymore available. So what we need is more or less a real good treatment. Nevertheless, what is the actual and current state of the art diagnostics? It's more or less you take your sample, for example, urine sample, then you do a cultivation. This takes up to 24, 48 hours. Then you can identify the bacteria, you know, gram-positive, gram-negative. Then you can go for the identification of the antibiotic resistance. This takes another 8 to 10 or 12 hours. This you can do, for example, with the VTech from Biophotonics, and then you can come up with a therapeutic decision. So what is needed at the very end, a very fast and precise method, which is just faster, much faster. And this is where we want to go in. We want to take the patient sample, isolate the bacteria out of this, and then just take spectroscopy together with microscopy to measure to identify the bacteria and to do the susceptibility testing. And then we developed a nice technique, which we call the RAMANBIOASSAY. It's a chip-based device which has been developed in our clean room. You have here on this chip something like 20 little containers where we can put the antibiotic in a lyophilized way, and then we put our bacteria into this. Then we have an interaction going on up to 90 minutes or 120 minutes, and we can continuously measure Raman data. We have some electrodes here, and why do we use these electrodes? Quite simply because then we don't need too many cells, because with these electrodes, if we have, for example, here plus and here minus 4 volt, and this is alternating, then we can push by dielectrophoresis forces the bacteria into the center of the cell, and then we can nicely measure them by Raman spectroscopy. And here you can see the containers. In the center, we have a hole where we have our control. This we also use for the identification of the bacteria. And around there, we have, for example, one type of antibiotic. And there we use normally 4 different concentration. And this means on this little chip, we can measure 4 different antibiotic at the same time with 4 different concentration. And we have 4 control measurements. Well, this is what it looks like in the scheme. So we have our 4 antibiotics here and here. Then we measure the microscopic information, because we can learn something about the morphology change due to the antibiotic, or we can take the Raman information, or not all, but we take also the Raman information. And this we can then combine, and in this combination, we can produce a qualitative and quantitative resistogram. What does it mean? Quantitative, for example, if it takes here this Escherichia coli at 90 minutes, and here you can see this is the typical Raman spectra. If you look here in this range, where we have the DNA bands as well as CC bands, CH bands, sorry, and then we can see here a change And out of this change, we can also get, for example, here the MIC, the minimal inhibition concentration. And we are working on an automative way. So you can see here our chip, and then we can automatically push the chip to the other positions. And this way we can, in a very short period of time, we can measure the Raman data. And then by using some robotics, this chip will be exchanged and another chip will come. And this way we can measure many samples in a very short period of time. And here you can see we are testing many different antibiotics because this is needed for a resistogram or antibiogram. And here you can see we have various antibiotics. We have our controls, and then we can see by the Raman, by the microscopic image. Once in a while we see changes. Then we have the Raman data, and then we can see, oh, there is an inhibition going on. No, this one, for example, is growing. There's an inhibition here is but growing here inhibition, and then we can also go for example, after 120 minutes. For the MIC, we can really say what is the concentration, and is this related to a sensitive strain or is it to a resistant strain? And as I said, just based on Raman spectroscopy. Just to compare this, so this is the device what we have developed and what we try also now to test even more in the hospital and going for also new chip-based systems. So if you compare this with the classical approach, this needs something like 16 to 24 hours. If you go to new machine approaches, they still need 6 to 12 hours, and our approach is around 2 to 3 hours, and also quantitative like the classical microbiological technique. Well, with this, I'd like to change to a little bit a different topic. Again, a mega trend in the healthcare or a major issue, and this is certainly related to the aging society. And since we are getting older, the probability that we get cancer is increasing. But also, I'd like to show you that with these spectroscopic approaches, what we are using here, we can also go for the bowel disease. And again, we can also go for dementia using spectroscopic approaches, as well as look at certain eye diseases. I have to check a little bit for the time. Just give me a reminder when I'm losing or using too much time. So in this case, since we are using or we want to measure larger areas, then Raman is still quite nice, but there are other techniques which are much better. And therefore, we talk about here a multimodal nonlinear microscopic approach. And here for this intraoperative imaging technology, and the techniques we are using is coherent anti-Stokes Raman scattering. For those who are not really familiar to this is a quite simple technique. You can use two lasers, pulsed lasers, you tune them to a specific vibration, then you can you produce something like a polarization grating, and this you can easily probe by a third laser pulse, and then you get the anti-Stokes signal. And this the advantage is that we are on the other side of the laser excitation, so fluorescence is not anymore an issue. For Raman, very often biological sample fluoresces, so you have to think about getting rid of this fluorescence, and we have different approaches developed, but here this nonlinear approach is more or less free of fluorescence. And when you use this approach, then can you also go for stimulated Raman, and since we are using pulse laser systems, you can also go for 2-photon fluorescence. You can go for FLIM, fluorescence lifetime, or for SHG, second harmonic generation. And by the combination of these modalities, you get also quite nice chemical, but also morphological information at the same time. Yeah, as I said, from ideas to instruments is somehow driving us, and this is one of our first microscope, what we have been tested in close to the operation theater in the hospital for head and neck cancer tissue diagnostics. And this was in 2014. Now this is 2019. You can see this is a nice miniaturized multimodal imaging device. And here you can see the strengths of Jena. This laser is coming from the group of Jens Limbert, Andreas Thünemann from the IOF and the Institute of Applied Physics. And this is a fiber laser approach. And what makes it quite nice, it's quite stable. We get picosecond pulses out of this. We can use it maintenance-free for 24 hours, 7 days a week. We have a nice imaging speed by something like 1 megapixel per second. And we can go to the diffraction limit with the spatial resolution. And this allows us to measure, for example, quite fast. In 2 minutes, we can measure 1 square centimeters of sample, and then we have full resolution of this. Let's give you one example, which we have investigated together with Erlangen and Jena. This is on the inflammatory bowel disease. And there's, for example, the Crohn's disease or ulcerative colitis. 2 of these inflammatory bowel diseases, and the classical approach is either endoscopically, and then you look at the Mayo score, and here, for example, you see this would be called the Mayo score 0. If you take a sample and you look at the histopathology, then you look for the architecture, for the chronicity, as well as for the activity. Both of all of them are here in this case 0. And what does architecture means? This is more or less how is the degree of the decryption distort in terms of density, in terms of shape and arrangement. And then chronicity is related to how many lymphocytes and plasma cells are introduced or have been infiltrated into the lamina propria. And the activity, then you look at the neutrophils, how many neutrophils you have in the lamina propria, and so on. And if you go to the various scores, this is Mayo score, the endoscopic score, Mayo score 1, 2, and 3. And here for 3, you have really bleeding. It's an ulceration going on. And if you look for the histopathological score, then you can see here all the grips looks quite nice. And here you can see already some distortion. And this is taking more and more, and then you can see, if you look very precisely, the lymphocytes and the neutrophils being infiltrating into the lamina propria. The question is, can we get this information just spectroscopically? Because all these findings, what we see here, they have no molecular information really being present anymore. So what is our nonlinear approach? What this looks like? For example, here, this was one of our first lab approaches. We were using here CARS at 2,815 per centimeters, 2 photon fluorescence at 458, and then SHG at 415 nanometers. And this is what our tissue looks like. You can see this is more or less healthy tissue. If you look very precisely, you can see nicely the groups. There's no distortion going on. You can't also find other cells. And if we now compare, for example, this healthy tissue with very diseased tissue, then you can see we get the same information by our multimodal approach. And we will be able to learn something about the architecture, the chronicity, activity, as well as on the Mayo score. And again, here with this Mayo score 3. But what we learned from our medical people, they are not really interested in learning those new techniques, because every day, more or less, a new technique is being introduced, and with new features, and with new images, and then there would be a great or large learning curve for them. So what they want to have is more or less, can we also get an automatic disease characterization? Can we translate this information into something what they know? And therefore, we put a lot of effort, especially the group of Thomas Bocklitz at the IPHD. For example, can we go for something like a non-pathological staining, where we don't have to stain the sample anymore, and where we have no annotation at the very end required? Certainly for the learning we need it, but at the very end, when we have taught our algorithms, then they don't need anymore this annotation. And then if this is possible, then we have a real-time disease assessment. Well, here we have used, for example, introduced the methods of the autoencoders, not really going into the details, but I'd like rather to show you what we can get out of this. This is here, for example, here, this multimodal patch approach. We have here one patch, another patch, and then we can look at the pathological annotation. This was one of the machine learning approaches. You can see the borders quite nicely. but also some misinterpretation. And with the deep learning approach, this autoencoders, you can see that we nicely can identify these patches. And if we go now for the MyoScore 3, again, you can see here pathological annotation, deep learning having more problem in this case, but with the machine learning, the deep learning, with the deep learning, we were able to really go for a good automatic annotation. Well, what is quite important at the very end, we want to translate this also to in vivo. We want to go for an in vivo optical histopathology. And this means at the very end, for example, for cancer diagnosis, we want to see where is the cancer, where's the healthy tissue, where's the diseased tissue. And this is what the endoscopic picture would look like. And then with our laser, we would scan, we'd like to scan over the surface. And then in an automatic way, we want to see where is the tumor, where's the tumor border, where it needs to be cut. And this we can do continuously so that we can also go deep into the tissue. Well, and this is what is anticipated. At the very end, we want to really overlay where is the healthy tissue, where is the diseased tissue, so that really the physician knows where to cut. Well, this is one of our fiber approaches, what we have developed. It's a little bit bulky, but it can be used easily in the field of head and neck cancer. This is a multimodal approach with a multi-core fiber approach. We have something like 10,000 core fibers being used for this. And certainly what you get is a different response of each of these fibers you have to take into account. And what is the approach? What does it look like? We have here this Fujikura fiber, 10,000 cores. Each core has a diameter around 2 to 3 microns, and we just simply scan over the back of this fiber. Then we produce at the very end of the fiber here, this at the head, our signal, and then in backscattering geometry, we collect the signal, and then we go on a single fiber, a multimodal fiber on a detector, and then we can recalculate the image. And we have the modalities. What we take is CARS, SHG, as well as 2-photon fluorescence. And we measure also the laser background for the calibration of the intensities. As you know, this nonlinear processes really depends on the laser intensity, so that we can have this always in mind. And you can see when we scan with the hand over the surface, so this is a real-time imaging, what we can do. The field of view is something like 300 to 400 microns, what can be imaged at the same time. And here you can see one of our probe images of head and neck cancer tissue. And here you see the LSM picture. Well, you see it's a little bit different, but this is due to the fibers and to the recalculation of the image. But you can see that we can nicely reproduce the LSM picture by our fiber approach. And this allows us really going to in vivo applications. Then we have been working on pig colon, because also we want to go with this approach for colon scanning, colon tissue scanning with a new probe and a new probe head, which is much smaller. And here you can see that we are also able to get all this feature, what we get in the LSM picture, inside also this fiber probe image. The resolution target in this way were some speeds. And you can see that we can nicely differentiate between 3 and 6 microns. So our resolution is around 1 to 2 microns, what we can achieve. And if we have a couple minutes left, not too much, then I'd like to introduce you something which is really fascinating, because this was the first time that we were able to use Raman spectroscopy directly on an eye. Yeah? And we want to use this eye as a window to the brain. And this is a European project together with the Technical University of Vienna. Rainer Leitgelb, Wolfgang Drexler are the PIs in Austria. And here we want to have an approach which allows us to really go for the retina, to learn something about the chemical composition of the retina. And here, this is what an HE stain would deliver to you. You can learn something about the nerve fibers, the ganglion cells, the interplexiform layer, the nuclear layer, inner and outer, and so on. And this is what normally a microscopic image looks like. And I'd like to show you later on that by Raman spectroscopy, you can get similar features, you can identify the various layers, and you can also see something going on. Is there any alteration going on? And this is, again, our motivation, but we want to use the Raman here together with the OCT and fluorescence. We want to learn something about the age-related macular degeneration, which is one of the major causes of blindness in Europe. And the other topic, we just published a nice paper there, is on Alzheimer's diseases. We want to use the Raman approach to see if there is something going on, some changes on the amyloid beta deposition in the brain, on the retina, because this will be seen as a part of the brain. And we have just been able to show on some mouse models, AD mouse models, that we were able to really nicely distinguish between the healthy and the diseased tissue. And this may be one of a new technique for an early access of this Alzheimer's disease. Well, this means the aim is we want to diagnose the disease at an early stage, and this in a fully operative manner. And here we want to combine OCT and Raman spectroscopy, but also then later on fluorescence. So here, this is the approach, the technical approach, OCT, and then we have also here a line field scanner, laser ophthalmoscoping included, Raman and fluorescence. And we want to scan over the retina. So this is the final goal, doing this in vivo. And I'd like to show you some of the devices or the development of what we have achieved so far. I'm not the OCT guy, I'm the Raman guy. Therefore, I'd like to talk about the Raman. And what you can see here is an isolated human retina. And this we scanned by Raman spectroscopy. And then we identified the various components. And what we can see here is that we have, for example, here in the center, we have localized carotenoids. We have also some of these here within the carotenoids. We have also an accumulation of lutein, zeaxanthin, and beta-carotene, as well as what we can see here is lipids, where we have, let's say, a lot of proteins and vice versa. And this is what such a typical spectrum looks like. If you look at our publication, then you will see that there is a wide, large spreading. We have a big fluorescence background, and we have used here this AMC approach in order to remove the background. And then we did a hierarchical cluster analysis to develop to see what are the various contributions. And here from the color, you can see where we have here the different contribution. And this is where we have these carotenoids here in the center. Then we have combined this with OCT. This is the OCT image. And from the OCT image, you can see there is something different, but it's hard to see the chemistry. You get morphological information, cell information, but really to see the chemistry is quite challenging. Nevertheless, we overlaid here now the Raman approach, and you can see here that we have a nice agreement, and we can find the various chemical compositions now here in the eye. Then we have developed the Raman spectroscopic approach in such a way that this is eye safe. So we have used the optical parameters for a normalized eye, and to be also to fulfill the ANSI limit. And therefore we are using now the spot size of 100 microns. We have a very low confocality. We have a very low numerical aperture. This is 0.47. Now you have to really look at the intensity. We have used just 1 milliwatt on the sample, and the laser light was 785. With these parameters, we were still able, not at the moment on a real eye, with the mouse, yes, but not on humans, but this will come quite soon. But we have mimicked a human eye, and you can see that we can now nicely see these are the Raman data. And you can see here the red spot here measured on this yellow spot. This is the white here, the white spectrum. And here you can see what is achieved with our home build setup. So these are under in vivo condition, fulfilling the ANSI limit. So this allows us now to go to the humans, and there will be in Vienna quite soon first measurement on the human eye. And here you can see the data which we can achieve with a confocal high-tech Raman microscope. And you can see that with our home built device, even with this very low density, photon density, we were able to get reasonable spectra in order to interpret all this data. Well, with this, I'd like to close. I hope I could show you somehow that with this research approach of translational biophotonics from ideas to instruments, that we are really able to go for the unmet medical need. What is needed? You need imaging, you need spectroscopic approaches, which are label-free. This is essentially because then it will be, otherwise it will be very difficult to go for in vivo. You need to be chemical sensitive, you need to get morphological information. So this means you need morphochemical information. You need to be fast and non-invasive in order to be able to characterize cells, but not one cells, but many cells, like for the host response. And if you want to go for larger areas like tissues, then you have the same requirements, but there we use, for example, now the nonlinear modalities. And what is quite essential in order to interpret the data, it's quite important that you are really going here for a combination out of biophotonic imaging and spectroscopic techniques with chemometrics, with imaging analysis. And chemometrics, I would say this is just the placeholder for everything what you know from machine learning, deep learning, artificial intelligence. At the very end, we need a sophisticated mathematical interpretation of our Raman data. Well, and this then allows you really to go for precision medicine. In this case, precision medicine means that you can really go for a target therapy on an individual patients and the patient's need. Well, with this, I'd like to thank the people doing all this wonderful work. And you can see we have PhD students, we have senior scientists working on these various topics. What is important, funding here for this type of research, and we need a lot of funding resources. As we have seen, one half of the funding needs to come from third-party funding. And what is quite important in order to do this research, you need good collaborators on the technical side, but also on the medical side. And we are quite grateful that we have in Vienna, but also overseas and in other cities, very good collaborations going on in order to really push these techniques to the border. And with this, I'd like to thank you for your attention.
Thank you very much, Jürgen. I'm going to applaud on behalf of the entire institute. So thanks for this frontline insight into the opportunities and challenges when you're trying to move biophotonic approaches to clinical application. Are there any questions? So far, we don't have any in the chat. If you want to ask a question, put a Q in the chat, please, so that we know that you want to ask something. And maybe I start. So, you made a point very early on that it's not easily possible to distinguish different cell types based on the Raman spectrum, because obviously you have, nucleic acids, amino acids, sugars, lipids, the same kind of molecules in every single cell. But is there a difference in the molecular composition, say, between bacteria or viruses or fungi that you could then use to distinguish them from cells?
Okay, so let's say from bacteria to a virus and to a cell, this is quite simple. This is the combination out of size as well as the chemical composition. Yeah, so this is quite simple to differentiate. But when you go from one virus to the other virus, it's already getting challenging, especially also with the size of these virus particles to do single virus detection with ordinary Raman is not really that easy. So what we use here is this tip enhanced Raman spectroscopy. And there we could show that we can easily then distinguish between various virus particles. For the bacteria and for the cells, at the very end, what you need is a supervised approach. This means you have to learn your algorithm. What does a normal neutrophil looks like? What does an eosinophil looks like? And this you have to train your algorithm, and then you can do the identification based on this classifier. And the same holds for the bacteria. So what we built there is a database of the most common bacteria, which is, for example, causing an infection diseases. And this is not too much. Normally you have something like 20 different type of bacteria causing this infection. So this means that you have just to make this database out of this 20 type of bacteria, let's say like E. coli or Enterococcus faecalis. Then you have to take some of the bacteria which are on your skin. Because if you have a catheter infection, then you have to distinguish between the bacteria coming from the skin versus the bacteria coming from the urinary tract infection. And this way you build your database, and then with this classifier you have built, you can go then for the identification of these unknown particles.
Okay. Thank you. So we have a few other questions now. KP, do you want to unmute yourself and ask your question?
Thank you, Dr. Popp, for the great talk. It was really nice. Yeah. So my question is regarding the eye applications. So you said like there will be about 30 seconds of integration time. Although we are using power of 1 milliwatt in the eye, but have you thought about like if there will be eye movements? Because if you measure it on the actual patients, there will be eye movements. So would you be using something like an eye tracker or something like that?
Not at the moment, because our spot size is already quite large. As I said, we are using something like 100 micron. Yeah, and if there is a movement of the eye, this is not really. If you go through this complete eye, yeah, then there is not too much movement on the back on the iris.
Okay, yeah, thanks.
But at the very end, we will see this when we do many measurements on humans. How this at the very end really looks like. What we can easily see when we go for in vivo detection of, let's say, for example, if we do an intravital microscopic approach, for example, if we look at the, I didn't show any of those results. If we look, for example, at sepsis, and if in the case of sepsis, a lot of, let's say, medicine is going, or biomedical is going on in the liver. And if we then do imaging directly on the living liver, then you can easily see there are motion artifacts by breathing, there are motion artifacts by the blood pressure, and this we need then really to take into account. And therefore we have to develop algorithms that we can stitch our images then really nicely together, not depending on these artifacts.
Okay, let's take 2 more questions. So Timon, maybe you could unmute yourself.
Yeah, thanks again for the great talk. I have a more technical question. So in the beginning of your talk, you showed that you do phenotyping of single cells and that you basically spread them over a chip. So how do you scan them? I mean, do you really just scan the whole chip, or do you have any mean of kind of picking the cell, and do you just then take 1 point per cell, or how do you do that?
So we developed this high-throughput Raman in such a way that we take a certain image, then we identify where the cells are located, and then we move with a fast table the cell beyond the microscope, and then we don't scan over the cell. What we take is an average spectrum. We have our focus widened up to 8 to 10 microns, and then we take 1 single shot spectrum, and then we move to the next cells. And the secret, more or less, is that we have developed also very sophisticated autofocus system, which allows us really to go for this large frame of this cover slide. Because otherwise, if you don't have autofocus inside, then you will lose the cells, because they are just something like 8 microns in height, and you can easily have a tilt by something like 50, 100 microns on your cover slide. And this you have to take into account.
Okay, thank you.
Okay, Florian.
Okay, thank you very much for the very interesting talk. I want to come back to the, say, machine learning-based identification of things based on the Raman spectra. And the question I have is, do you have the feeling that you are already, with this technique, extracting, so to speak, the maximum amount of information that is in these spectra? And, so to speak, the error that remains you have to live with, there is simply nothing better, simply because there is not a one-to-one correspondence? Or is there still room for improvement by having a better neural network, paying attention to very fine details that maybe now are not looked at?
Right, so this is really a very, I would say, challenging question, because at the very end, there is not a real gold standard, yeah, we can compare with, yeah. And this already starts, for example, if we look at the different leukocytes, yeah, the neutrophils, and so on, then when we get a sample which is mainly pure, this not necessarily means that it's really 100% pure. Yeah, it is always some contribution of other cells inside. Yeah, and therefore we already tackling with our, let's say, gold standard. Yeah, what is really the ground truth? And what we realized is, yes, with the more we learn about the mathematics and the statistical approaches, and as well as going from, let's say, from the classical machine learning chemometrics towards the deep learning, if the set of data is large enough, yeah, and then we can improve our findings. But what is not really satisfying is when we go to these really deep learning approaches, or AI, so to say, then the understanding is getting more and more lost. And therefore, we want to go for an interpretable AI so that we can look at the hidden layers and can understand why was the decision this way and not the other way around. And to be honest, I'm pretty sure we are just at the tip of the iceberg what mathematics and statistics can do. I'm pretty sure there is more room left.
That sounds good. Thank you.
Thank you.
Okay, time is quite advanced, but there's one, maybe a short technical question by Masood. He's asking which wavelengths are you using for Raman devices, and how do you get rid of the fluorescence signal that you mentioned? So maybe a brief answer on that.
To be honest, we are using all wavelengths which are available. What I have shown here for the cells, this is 785. And in order to get rid of the fluorescence, we have approaches where we have this multispectral analysis where we can really go and fit the background and take this out. If we go into the UV, then this is normally background free. But there we have the problem that this UV radiation may, let's say, alterate our sample, and therefore we need to rotate in order to have the photo degradation really not being affecting our Raman results. So to say, we have all lasers we are using depending on the question we want to answer. And for the cells, it's 785, 830 nanometers. For the bacteria, we go for 530 nanometers. Why we don't stick there with 785? This is always a trade-off with the power to 4 dependence of the scattering. Yeah. So if you go more into the red, you have to integrate longer in order to get the same amount of photons. And therefore, we want to go always a little bit more into the visible in order to collect more photons in a short period of time.
Okay, so thank you very much again for this great presentation of your research and the discussion. Thank you very much for joining us today. You're aware of the fact that we now have these very individual meetings, but before I let everyone else go, there's 2 things I wanted to say. This was...
First, many thanks for listening. And as I said, I will be around for the discussion. And so I will leave for a quick bio break, and then I'm looking forward to having some nice discussion going on. Thank you. Bye-bye.
In a minute. So for everyone else, since this is the last series, the last seminar in this series, I wanted to say that we're starting a new series, and it will probably continue in April. And Lothar Kuhn has just posted some information on this. When you're interested, if you're interested in being on top of the latest information on that, you can sign up for a newsletter, and then you receive information on what seminars we're planning and when they would occur. This is also for our external guests. So this was point number one. And point number two is that, again, Lothar Kuhn, as the head of our PR and interested in communications, suggested that we do a little survey at the end of this series, just to get your opinions about what you think of the DLS seminars in this series and what we can do to improve. So I invite you to participate in this poll here.